Assalamualaikum, welcome back to my video. In this video, we will discussing the detailed step of meiosis. Before we discuss more on detailed step of meiosis, let's take a look the important events between the nucleus splitting, which is the interphase. So interphase is the preparation stage for meiosis process that consists of G1, S and G2. The chromatin in the nucleus will start replicate themselves in the S phase, the process known as DNA replication, but the chromosome cannot be seen individually because they have not yet condensed, and the two centrosomes formed by duplicating of a single centrosome. Each centrosome contains two centrioles. Now we move to the first division in meiosis, which is the meiosis 1. So meiosis 1 contains prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis. During prophase 1, the nuclear envelope and nucleolus starts to disappear. Chromatin slowly condense into chromosome. The chromatin tightly coiled, they become shortened and thickened. And then microtubules grow out from a pair of centrosomes and they migrate to the opposite poles of cell. Homologous chromosomes begin to pair up in process called synapsis. So now the paired homologous chromosome, we call it as bivalence because they have 4 sister chromatids. And then during prophase 1, there is important event which is the crossing over. So crossing over occurs during prophase 1. The non-sister chromatids exchange their DNA segments. And then the place where crossing over occur, we call it as chiasmata. So chiasmata is the X-shaped region where crossing over occurred. So crossing over begins very early in prophase 1 as homologous chromosomes pair up gene by gene. In crossing over, homologous portions of two non-sister chromatid trade places. Crossing over produces recombinant chromosomes which combine DNA inherited from each parent, from maternal and from paternal. Crossing over contributes to the genetic variation by combining DNA from two parents into a single chromosome. So next is the metaphase 1. In metaphase 1, homologous chromosomes line up along the metaphase plate. Both homologous chromosomes still attached to the another at chiasmata. The centromere of each pair of homologous chromosomes become attached to the spindle microtubule at the kinetochore. And then the next one is the anaphase 1. So during anaphase 1, homologous chromosomes separate. Sister chromatids remain attached to one another at centromere. No centromere breakup, no centromere division. Only the homologous chromosome have become separated. And then during telophase 1, chromosomes reach at opposite poles. The nuclear membrane reappear, followed by cytokinesis. So cytokinesis takes place before cells enter meiosis 2. In animal cells, a cleavage furrow forms. In plant cells, a cell plate forms. So no chromosome replication occurs between the end of meiosis 1 and the beginning of meiosis 2 because chromosomes already replicated before meiosis 1 in interphase. So in meiosis 1, the two haploid daughter cells form and each chromosome still consists of sister chromatid whereby they still attached by the centromere. Now we move to the second division in meiosis, which is the meiosis 2. So meiosis 2 contains prophase 2, metaphase 2, anaphase 2, telophase 2, followed by cytokinesis. During prophase 2, the nuclear membrane disappears and the microtubule form. Each chromosome consists of two chromatids and still attached at centromere. Synapses do not occur as well as crossing over do not occur. And then metaphase 2, each chromosomes line up in the metaphase plate of cell. The two sister chromatids of each chromosomes are not genetically identical. The kinetochores of sister chromatids attached to the microtubule that extending from opposite pole. And then next, during anaphase 2, the sister chromatids separate. There is a division of centromere. Centromere breaks up. And then sister chromatids of each chromosome now move as two newly individual chromosomes toward opposite poles. And then the last step is the telophase 2. Each chromatids reach the opposite poles, nuclear membrane reappear, microtubule disappear, followed by the cytokinesis. In animal cell, there is a formation of cleavage furrow, while in plant cell, there is formation of cell plate. Thus, meiosis division produces 4 haploid daughter cells that have the variation in their genetic content. There are three mechanisms that contribute to genetic variation during meiosis. Number one is through independent assortment of chromosomes. Homologous chromosomes oriented randomly at metaphase 1. In independent assortment, each pair of chromosomes sort maternal and paternal homologous into daughter cells independently of the other pair. Maybe the chromosomes line up in combination number 1, or maybe combination number 2, number 3, or number 4. The second mechanisms that contribute to genetic variation is through crossing over. So crossing over produces recombinant chromosomes which combine DNA inherited from each parent. And then third mechanism that contribute to the genetic variation is the random fertilization. So fertilization occurs subsequently after the production of the gamete. So random fertilization adds to genetic variation because any sperm can fuse with any ovum. There are some important stages in meiosis, which is prophase 1. Each homologous pair undergoes synapsis. Synapsis means they pair each other. And then crossing over occurs between non-sister chromatid with the subsequent appearance of chiasmata. And then during metaphase 1, chromosomes line up as homologous pairs on metaphase plane. And then during anaphase 1, homologs separate from each other, but the sister chromatid remain joined at the centromere. And then there is some differences of mitosis and meiosis. For example, mitosis occurs in the merismatic cell and somatic cell, while meiosis only occurs for reproductive cell, it producing the gametes. And then the number of chromosomes of daughter cells is similar to the mother cell. We call it as diploid in mitosis, while in meiosis, the number of chromosomes of daughter cell is half of mother cell. We call it as haploid, represented as N. During mitosis, in late prophase, homologous chromosomes do not synapse to form bivalent, while in meiosis, in late prophase 1, homologous chromosomes synapse to form bivalent. And there is another difference of mitosis and meiosis. You need to remember at least 5 differences of mitosis and meiosis.